Good evening, everyone present here. Myself, Shouradip Odhikari. On behalf of Modok Trainer Academy, I extend a very warm welcome to all participants joining us today this important and timely webinar on role of microbes to protect the environment. We are truly grateful to each one of you for taking the time to be with us this evening. In recent years, conversations around environmental protection have become more urgent than ever. Microbes play a very crucial role in maintaining ecological balance, cleaning pollutants, and supporting sustainable life on Earth. It's a matter of great honor for us to have Dr. Srabanti Basu with us today as the distinguished speaker. Dr. Basu is a professor and the head of the Department of Biotechnology at Heritage Institute of Technology, Kolkata. She has completed her MSc and PhD in biochemistry from the University of Calcutta and has extensive research experience in bioprospecting and bioremediation. She is also a recipient of the prestigious Fulbright Nehru Fellowship and several national and international awards like Young Scientist Award by Indian Science Congress Association 1999, International Conference on Sustainable Green Frontiers 2025, and many more. She has 4 book chapters and 29 papers in international journals to her credit. With decades of academic and research experience, her insights will be extremely valuable for all of us. So, without further delay, I cordially invited respected Dr. Srabanti Basu to take over the session and guide us through this fascinating topic. Thank you.
Thank you. First, I like to know whether my voice reach you clearly or not. Um, can you
Yes, ma'am.
hear me? Okay. Before starting, I must thank Modok Trainer Academy for inviting me to today's program as a speaker. Uh, I want to speak on role of microbes to protect the environment. So this is a very crucial topic. Yes, as it was rightly mentioned, that environment is a very important part, because if we don't have our environment protected, then our existence will be at stake. So can you please share the slides? Because yes, thank you. So the role of microbes. My topic is role of microbes to protect the environment. Next slide, please. Before going to the microbes, let us see what environment is. So we all know that our surrounding is our environment. So, but there must be some definition. So there are two models for defining the environment. The first one states that the part of the world or the part of the earth which is without human civilization is called environment. That is, excuse me, environment is something which is not touched by human civilization. According to this model, wherever we have human civilization, environment is lost. Now the question is, if this model is accepted, then there is no environment left on the earth today, because everywhere we have human civilization except some few pockets. So if we don't have environment, then where does we get the question of protecting environment? Environment is already lost. So another model was proposed. Next slide, please. According to that model, human civilization is an integral part of environment. In human civilization or human settlement is a part of environment. So it is not that human destroys environment. Instead, human, according to this model, or is a um, is a citizen of environment, is a citizen of the entire world. So humans are responsible to protect the environment. They have the they have the responsibility to protect others. And every citizen of the environment has has the that they have the right to exist. So it is our duty to protect all the citizens of the environment to keep the ecological balance. It is a necessity. Next slide, please. Now, another basic question we will have, another basic point we will discuss, that is how big the environment is. Basically, there is no limit. We can consider our environment a watershed, a grassland, pond, everything, river, a playground, everything can be an environment. The room where I am sitting in, that is also has an environment. So it is extended everywhere, and it can be extended to a state, to a country, to the biosphere, and also to the universe, because the extra um, I mean the cosmic environment can affect the earth also. So, and there is no limit of environment. We cannot say that this is environment or that is environment. And another important point is that there is no absolute environment. This watershed is an environment. We cannot state that. This is another environment. We cannot state that. So there is no absolute concept of environment. The concept of environment is relative. This is my environment. You can have your environment. You can have, or he or she can have his or her environment. Like this, a tiger will have its environment, a bird will have its environment. So everybody has its own environment. And so it is in a collective manner, we have our environment. That is very important. Next slide, please. So now we will come to the role of the, okay, we can skip this slide. Next slide, please. Now we will come to the role of the microbes to protect the environment. The microbes have different roles to protect the environment. Now, what happens? The environment changes the humans uh, mm, due to their development, and for their development, for different types of developmental projects can change the environment unwillingly, unintentionally. That is, they are doing something for their own development, but that is affecting other members' environment. So that is some unintentional changes humans have made. And gradually we are moving towards a human centric civilization. But it is very um, uh, very unfortunate that we are not looking after others. Now, that is another issue, but the point is that we have gradually changed our environment. Now, but if we change our environment, then the problem is that the ecological balance will be lost, because everything is connected with everything else. And if we cut a point, then the entire network will be ultimately can get devastated. It can tolerate up to a certain extent. Then after that, it will get devastated. And that ability of the healing nature, the self healing, that property of self healing of nature will be gradually lost after a certain extent. And we have touched that, we have come up to that limit. Now, their role, microbes play a very significant role to protect the environment. There are two different aspects. One is that it can maintain the balance of the environment, the microbes, and the other part is that they can be utilized to make a better environment, to make the environment, to keep the environment balanced, or they can be used for some technological purpose so that we can reduce the use of chemicals, we can reduce the use of energy, etc., etc. So, what does actually microbes do? I will focus on the second part, that is how the microbes can be utilized to keep a better environment. Now, when in the last century and also last to last century, in the second half of the 19th century, since the second half of the 19th century, we used a lot of chemicals and human development took place very rapidly. And there was a chemical age, we can say. And the pollution was so high and it gradually became visible at the end of the last century. And so the microbes were exploited to for alternative processes by which we can uh, mm, reduce the use of chemicals. Then microbes can be utilized for reduction of greenhouse gases, or they can be utilized for sources of as sources of bioenergy. Then microbes can be used as biofertilizers and they can also be used for removing pollutants. Next slide, please. Now let us see what are microbes. Um, by microbes, we mean the tiny organisms which we can see only through microscope. We have bacteria. By microbes, we mean bacteria, fungi, algae, and also viruses. But viruses today, in today's discussion, we will not consider the viruses because they are not actually utilized for the beneficial purposes. Instead, we use different bacteria, fungi, and algae for protecting our environment. Next slide, please. Now let us come to a very old process for using microbes for alternative productions, alternative pathways. I will start with an example of acetone and butanol production. We can produce acetone and butanol by bacteria. Initially, it was produced by by a chemical process, which needs high temperature, and several chemicals were required for. There were several steps, and several chemicals were required. Now, Louis Pasteur in 1862 first reported that microbes can produce acetone and butanol. And he actually reported butanol. Then, side by simultaneously, acetone was also produced, and he named the culture Vibrio butyric. So later it was found that it is not a pure culture. That means a single organism is not present. There are several organisms. There were several organisms in that culture, and it was found that it was suspected that at least one was Clostridium strain. So there is a particular bacteria, Clostridium acidobutylicum, which can produce that butanol. It was first reported by Pasteur, but it was kept aside. Now the first commercial interest developed at the beginning of 20th century. There was a need for production of chemical rubber. And then Weizmann isolated a new bacterial culture, and then that was Clostridium butylicum, and that could ferment, um, that could produce butanol and acetone rapidly through fermentation. And also, very little amount of chemicals were required. And most importantly, the process came down to the ambient temperature. So energy requirement was low. Now, during the Weizmann, that process was developed in 1914, and in 1915 a patent was granted. And that is the process we still use today. By production, we produce butanol and acetone using Clostridium acetobutylicum. And thus we could reduce the use of chemicals, we could reduce the use of we could reduce the lower the energy. We could do the process in the ambient temperature. Now, the requirement or the demand of acetone increased during the First World War. Again, that time butanol was not was not considered very important. Acetone was required for producing different war chemicals. Now, after the First World War, the interest went off. Again, when the Second World War started, people became interested in acetone production. Again, after the conclusion of the Second World War, acetone production, butanol production were stopped. And that time, butanol was considered a byproduct, an unwanted byproduct. That is, there was no need to have the byproduct, but it was there. It came as byproduct, so it was simply stored. Now, after that, just before the First World War, automobile industries developed, and then the interest in butanol raised because butanol was used as a solvent for the lacquer-based paints. So people started producing butanol with Clostridium acetobutylicum, and then again with the Second World War, the butanol and acetone production took place simultaneously, and the people were interested in both of them. Then again, the thing that interest subsided. But after 1970s, and particularly at the beginning of this century, this interest again came up. And that is for a different reason, because butanol is now considered an alternative fuel. So we can use this bacteria for production of butanol, and which can be used as an alternative fuel. Next slide, please. This just shows I just put this side to show the biochemical pathway by which we can use, by which the bacteria can produce acetone and butanol. There are several steps, but all take place within the cell, and they all take place in the bacterial temperature, that is the ambient temperature, and there is no need to add additional chemical. This conversion, starting from glucose, this conversion can take place within the bacterial cell. So we can just add glucose to the bacterial culture. The bacteria will take up that glucose, and that will convert glucose to acetone and butanol. Next slide, please. This is now the point is that we are interested in biofuel. So that is again the bacteria or the microbes can solve that problem also. That is, we can use carbohydrates, lipids, lignocellulose, etc., and or cellulose, etc. So these, sorry, starch, cellulose, lignins, lipids, etc., we can use, and those can be converted to butanol as well as ethanol, isopropanol, etc. They can also be converted to other things, that is mostly isopropanol, butanol, hexanol, and they can also be converted to octanol. And all of them can be used as biofuels. This bacteria can be used for biofuel production. And there are several organisms like Clostridium acetobutylicum, Clostridium thermocellum, E. coli, then Pseudomonas putida, Zymomonas mobilis, etc. They can be used for production of these alternative fuels.
Hello, ma'am.
Yes.
Yeah,
So we can. use these fuels. Now, what is the problem with the normal fuel or conventional fuel? That is, I think it is quite well known to all of you that the conventional fuel, we now use the fossil fuel, the petroleum and the coal, and it is gradually we are using up the sources, and the source is gradually declining. So there will be one day there will be no fossil fuel, and so they cannot be renewed or they cannot be replenished. Now, this type of, if we can produce this type of bioethanol, ethanol, I mean ethanol, butanol, etc., by fermentation, then they can be mixed with the conventional fuel, and we can thus reduce the use of the conventional fuel. They can be normally used as the car fuel, automobile fuels, etc. So, but they cannot be used 100. You cannot use 100% butanol in an automobile system. You have to mix it up with conventional fuel. Um, Next slide, please. Now, in the next slide, they can also, microbes can also be used as the greenhouse for production, I mean reduction of greenhouse gases. There are some microbes which can utilize methane. They can take up methane and they can convert it to carbon dioxide. We call these microbes methanotropes. And these methanotropes can be used to reduce methane, to reduce the greenhouse gas. Now, carbon dioxide is also a greenhouse gas. So, if methane is getting converted to carbon dioxide, so basically the problem is not really solved. So we can use algae, microalgae particularly for this purpose. And microalgae can sequester carbon dioxide. So if we can grow, if we can make a co-culture of methanotropes and microalgae, then that will be a very good solution for reduction of greenhouse gases. Because algae will take up that carbon dioxide, it will convert it to glucose, and gradually glucose will be utilized by it, by the algae for its growth, and carbon dioxide, and it will release oxygen. So that carbon dioxide and water will be converted to glucose by photosynthesis, and oxygen will be released. So it is a fantastic system to reduce the greenhouse gases, and in many places people are working on this, and this type of uh, mm, uh, this combination is installed to reduce the carbon footprint and the greenhouse gas. Next slide, please. Now, microbes can be used as different sources of enzymes. They can produce amylase, cellulase, pectinase, protease, lipase, etc., etc., and they can be used for different industries like food industry, textile industry, paper, detergent. I have mentioned a few pharmaceuticals like that. So why these enzymes are beneficial? Because the process can take place at room temperature. We don't need to go for higher temperatures, and we can reduce the use of chemicals. Like one point is that just I want to mention one interesting thing, that is how you can reduce the use of chemicals. In textile and paper industry, we need polishing. That is the paper, we need to get polished papers, we need to get polished fabric. So for that, early days, they were treated with sodium hydroxide. And just by treatment with sodium hydroxide, the rough edges of cellulose were removed. Paper and cloth fabric both are made of cellulose, particularly the cotton fabrics, and most of the fabrics are made of cellulose. So the rough ends of the cellulose are removed by sodium hydroxide. Nowadays, sodium hydroxide treatment is not, I won't say is not done, it is done, but it is getting replaced by cellulase treatment. And microbes can be excellent sources of cellulase, particularly the fungi. Then this way, in paper and textile industry, you can reduce the use of chemical. Sodium hydroxide is getting replaced by cellulase. In textile industry, this use of cellulose has come is has become very prominent, but in paper industry, sodium hydroxide is still in use. Now we have the food industry, we have pharmaceutical industry, then we have food, pharmaceutical, and etc. In detergent industry, in detergent also earlier we used to have very harsh chemicals, but now we can use just um, detergent, which is the sodium salt of some petrochemical compounds. These compounds are biodegradable, these detergents. Along with that, we can use some enzymes, and we don't use to you don't need to use very harsh chemicals. Thus, we reduce the requirement of energy, and we also reduce the use of chemicals. Next slide, please. Now let us come to the biofertilizers. Biofertilizers are the we know that by using fertilizers, the soil fertility is gradually lost. Now, the concept of biofertilizer, it has become very important nowadays, but the concept of biofertilizer is not really new. It is here for centuries. So, in early days, people used to spread different biomasses over the like algae, dried uh, marine biomass, etc., particularly marine algae that was used by different locals in different by locals of different areas to increase the fertility of the soil. But the mechanism was not known. In 19th century, actually in 1895, the late 19th century, a biofertilizer was introduced in the market that was dried rhizobium, and the name was nitrogen. It was marketed, and before that, it was discovered that rhizobium can increase the soil fertility by increasing the soil nitrogen, by fixing aerial nitrogen into soil. But after that, the popularity was lost. I mean, popularity of that nitrogen was lost because chemists synthesized ammonia and other nitrogen compounds, urea, in their laboratory. So people were not interested in buying nitrogen, a bacteria. Instead, they could buy that ready chemical and they could apply in the field. But again, application of these chemical fertilizers actually reduce the soil fertility because the microbes gradually, um, the microbes of the soil, the natural soil flora, gradually die if we don't have organic compounds in the soil. So use of inorganic materials. Urea is not inorganic, but urea is converted to ammonia, and so it practically becomes inorganic. So use of these inorganic compounds actually decrease the organic compounds of the soil, and thus the soil flora is lost and soil fertility is lost if the organic compounds are decreased. So um, we we become totally dependent on addition of these compounds to the agriculture becomes totally dependent on the addition of these compounds to soil. So what happens when um, mm, uh, these organic so uh, the soil fertility to regain the soil fertility, we can give the organic compounds to the soil. But the point is that biomass is not there in the soil. So if biomass is not there, if microbes are not there in the soil, who will utilize those organic compounds? So who will convert the organic compounds to the nitrogen, to ammonia, so that the plants can take up? So that is for that gradually the biofertilizers become more biofertilizers regained their importance, and people again try to become interested in making the biofertilizers by utilizing different microbes. So we can use bacteria and fungi as the biofertilizers, and these biofertilizers can do several functions. On the basis of their functions, they can be divided into different categories. And they can just mentioning the functions, they can fix the nitrogen. That is, they can take up aerial nitrogen and they can put it in the soil. And they can secrete different hormones. They can solubilize different metals, and they can produce some compounds which can bind with some metals and which can transport different metals to make them available to the plant. So this way, they increase the fertility of the soil. Can use this bacteria to increase the fertility of the soil. Apart from this, several bacteria have been identified which have these properties of fixing nitrogens, enhancing availability of different minerals. Apart from this, they can survive in under some extreme conditions, like under salt tolerance, under high salt concentration, under at a very cold temperature, etc., etc. So they can be they so these type of bacteria can be used as biofertilizer to for different spatial areas, like salt. If we have salt tolerant bacteria which can fix nitrogen, then we can use those bacteria as biofertilizers in our say our Sundarban areas, in different marine areas, where we can cultivate paddy and other crops. So that is um, a very good prospect by which we can protect our environment, and also we can increase our agricultural um, yield. Next slide, please. So now we will go to the last part, that is how we can use microbes to protect, to clean up our environment. Where the environment is already polluted, how these microbes can be helpful to clean that polluted environment. That process is called bioremediation. We know that remedy means cure. So remediation means that is the curing of a site, or to go back to the normal site, to the normal condition or the original condition of the site by removing the pollutants from the site. Now, microbes. There are some pollutants which are called persistent organic pollutants. As the name indicates, that these pollutants are very difficult to degrade. They are organic compounds, organic in nature, and really they are difficult to degrade. They persist for a long time. Um, and there are some metals which have very high molecular weight, and many of them are quite toxic, like arsenic, cadmium, lead, chromium, and mercury. They are highly toxic, and all they are called heavy metals, and because they have high molecular weights, and the toxic metals are mostly heavy metals. Only one light metal is toxic, that is aluminium. Other than all toxic metals are heavy metals. So microbes can also sequester those metals. These metals cannot be removed from the site, but microbes can be utilized to take up the metals. But mostly microbes are used for the degradation of the organic pollutants. Now let us see why these pollutants are persistent, why they cannot be degraded by microbes. So if we look at their structure, we will see that they have fused rings, and many of them have at least one benzene ring, or and more than one benzene rings. They are mostly the polyaromatic hydrocarbons, and they have several substitutions. They are toxic in nature, And most importantly, the microbes did not have enough time for evolution to make the enzymes for degradation of these compounds. If we consider protein or carbohydrate, then microbes could get millions of years to live with these um, complex compounds, with this, I mean, with these complicated compounds, with these very big compounds. But petrochemicals were discovered only in the middle of the 19th, uh, 19th century, and its use has become um, it we used it in the beginning of the 20th century. It is for general, it came into the general use. So microbes did not get 100 years, did not even get 100 years to uh, evolve its machinery to degrade the petrochemical compounds. And these petrochemical compounds, the persistent organic pollutants, they are quite toxic. If they persist in nature, if they enter the food chain, then they will start inhibiting our enzymes. They will, they can bind with different proteins. They can interact with DNA molecules. They can go inside the DNA molecules. And when they interact with the DNA molecules, that means the interaction with DNA molecules means they are carcinogenic. They will cause mutation in the DNA, and that may lead to cancer. And they can also cause a particular type of damage which is called the oxidative damage. If we once the oxidative damage starts, it damages the entire cell, it damages the nearby cells also. Now some bacteria are there which live in the polluted area, and they are forced to live, they are forced to go with that those organic toxic compounds. So they um, have some enzymes for degradation of those compounds because they have those compounds as food, and they when they utilize those compounds, they have to, if they have to utilize the compounds, they have to degrade them. So they have some enzymes for degradation of those compounds. These bacteria are special bacteria. They cannot be found anywhere, and we have to check the soil. We have to go from site to site to get these bacteria. So they can be, we have to search for this bacteria. Next slide, please. Now, I will discuss one case study, how this bacteria can be used for degradation of the pollutants. This is with a petrochemical oil spill. If we have petrochemical spill, oil spill, or if we have site polluted with petrochemical degradation, then we can use this bacteria. So in this is one of the earliest, or I won't say one of the earliest, it is the earliest case where we used microbes to treat the bacteria, treat the oil pollution. So it was in 1989, March, on 24th March, the oil tanker Exxon Valdez that ran aground on Bly Reef in Prince William Sound, Alaska. That is an area of Alaska, and an estimated 11 million gallons oil spill. So that got sprayed as a surface lake over the ocean. Next slide, please. So this is the picture of that Exxon Valdez oil spill. The map that on day four, it was that sorry, it took place on the Valdez, that Exxon Valdez, that tanker was there on 27th March. Then on 30th March, after 7 days, around 9 miles away, it was detected. So it spread it gradually over a large area. Next slide, please. So gradually, within a few days, it was found that 778 kilometer of 4,800 shoreline of in Prince William Sound and 1,309 kilometer of the shoreline of Gulf of Alaska was covered with that oil. That was patchy, and the oil was like tar balls. So it was one of the biggest disasters of oil spill. Next slide, please. So what happens after that? EPA was consulted for that, and Exxon, and that is Exxon was the oil tanker. Exxon and EPA both suggested bioremediation. Exxon was the company. They both suggested bioremediation, that microbes should be applied, and along with the microbes, so they did the lab tests and they did the field trials, and field trials showed that there is a need of fertilizer. This fertilizer is not really the agricultural fertilizer. These fertilizers are nutrients which are required by the microbes for their growth. So what happens when so the field trials showed that there is that people need to add the microbes, and also people need to add the fertilizers, that is the nutrients, to um, get the best results. And at the beginning, there was 1.2% loss of hydrocarbon. Now the rate decreased when easily degradable compounds were consumed. And it was found that if the nitrogen concentration is increased, nitrogen by hydrocarbon ratio is an important factor for the rate for the biodegradation. That is, if you increase the nitrogen content, it will go high. So there was a need to add nitrogen salts from outside, and the time, gradually, if you increase the time, it will uh, that will that will I mean the biodegradation will be increased. Next slide, please. So in 1990, large scale fertilizer was applied, and it included 1,400 individual sites and 380 shorelines. And so it was, and it actually started in 1989, at the end of 1989, but in 1990, the large scale fertilizer application took place. In 1992, so after 2 years, a survey showed that most of the oil had been cleaned up. And on 12th June 1992, the US Coast Guard and the state of Alaska officially declared the cleanup concluded. So almost the cleanup was concluded, but it is not that all the oil has been removed. Next slide, please. So some oil was still there, and that is left for natural degradation. That is, the microbes which are present in the water, it was left for them to degrade that residual oil. In Finally, in 2001 and 2003, that is almost after 10 years, the National Oceanic and Atmospheric Administration conducted random sampling, and then they concluded that the ocean is now, or the site is now almost free from pollution. So it was done by the microbes only. Microbes were applied. Earlier, if we had this type of oil spill or oil slick, then they were then different surfactants and chemicals were used, by which the pollutants were allowed to spread over a large area of ocean, and they were actually remediated, or the site was cleared by dilution. So no degradation took place earlier. No degradation method was there. It was only possible because of these special microbes. Next slide, please. These are some actual pictures of the cleaning up processes. The first, that is physical washing of the shoreline, so with high pressure water, so that most of the oil goes to the water, and then the fertilizer was applied, microbes were applied, and then it was left for degradation. Next slide, please. In India, we have, not in India, I mean all over the world, including India, we now have different types of combinations of microbes which can be used as oil degraders and petrochemical degraders. And these are called microbial consortium, that is a formula of microbes. They can be applied for degradation of these petrochemical compounds or this polluted site, for remediation of the polluted site. Oil Sapphire is such a consortium developed by India. It was developed by TERI, and it has been successfully used since 1996 for removal of pollutants, and more than 285,000 tons of oil soaked or oil sludge, I mean contaminated slide, site, soil and water has been bioremediated by this oil sapper. So different beneficiaries are there, and it is has been developed by TERI. So and it is a very important consortium. That is why I mentioned it. Apart from this, different other consortia are also available. We will now, before concluding, I will show some pictures of this application of oil sappers. This is a petrochemical industry site where oil zapper was applied. Next slide, please. This is a petrochemical industry site where oil zapper is applied. The first, the top left, that shows the actual scenario of that the situation when the oil zapper was first applied. The top right, that picture shows the after the second application of oil sapper, the site has been almost cleaned up. Now the boundary walls, that is after the second completion of the bioremediation process, we can see the bottom right, which is the which shows the scenario after completion of the bioremediation. So if we compare the first application and the last slide, that is the top left and the bottom right, we can see the difference with the application of the bacteria, how the site can go back to the a site can be cleaned up. It went back to the original uh, condition. Next slide, please. This is another one that is in Duliajan, Assam. And the side, the top left, that is before application of oil sapper. The top right, that is the application after the application of first application of oil sapper. The bottom left, that is after the second application, and the bottom right after the final application or the completion of the bioremediation. So gradually, again, if we consider the first figure and the last figure, we can see the difference. And that is done by the oil zapper. Next slide, please. This is an application of oil zapper in Kuwait. You can see the change. It was the left one is the before the application of oil zapper. The right one is after the application of oil zapper. So to conclude, we can say that microbes, we usually think microbes cause disease whenever we say bacteria. Obviously, immediately a disease comes to our mind. When we say algae, immediately something which is we think something which is very unwanted and which is very disturbing. Fungi, again, something which cause diseases. So, and which spoil food, etc., etc. But basically, these microbes are very important components of environment, and they are tiny brains. So we should not ignore them. And with the microbes, we can protect our environment, and they are very, very important to keep the wave of nature intact. So that is, and we can utilize them also. We can utilize them, we can use them to clean up the environment, to protect the environment. We can take different avenues to reach a cleaner, to uh, reach a cleaner product, to reach a cleaner future. Next slide, please. Thank you for your patient hearing. Uh, thank you.
Thank you, madam. It was most fascinating and impressive session. Moreover, so important role of microbes for conservation in our life. So nicely, each and every slide full of information. So, and how does it work? So really nice. Thank you. Thank
Thank
you. Now I will just request our advisory board member, Professor Monotosh Dasgupta sir, to conclude the session.
Good evening, everybody. I thank Dr. Srabanti Basu for delivering this enchanting, catching, important, and knowledgeable account of the microbes and their role, different role rather, in case of maintaining the environment and also its use in manufacture of some very important chemicals. Practically, it is an ocean. And what she has presented is rather a small wave of it, because millions of microbes are there. Some are known, a large, pretty more number is unknown, yet to be explored. And this chemistry of microbes, it again consists of a number of realms. One, this chemical application. One is characterization of the microbes. Third, the nature of the microbes. If there is any alternation of generation, to identify that, so on and so forth. Practically, it comprises a number of subjects embedded together. We know the application of microbe in conversion of one chemical to another. In some cases, the mechanism is known. In some cases, the mechanism has not yet been explored. It is yet to be explored. And thus, it is an ocean. And many branches and sub-branches are there. And many scientists, they are working in their own sphere where these microbes are involved. Dr. Srabanti Basu has cited a window rather, through which we have had a glimpse of that ocean. Many more things can be known, many more things can be said. Therefore, I expect that in some future kindred lectures, he would explore some other sides of the subject. And again, viruses has been left out. He has, at the beginning, she has said that I am not discussing viruses. These viruses, role of viruses, that is again another big subject. Therefore, it is a fascinating subject. The glimpse of that we have had this evening again depicts how vast and how fascinating this subject is. I convey my heartiest feelings on my personal behalf and on behalf of my academy for having given us this idea that this subject is a vast one. This small glimpse we have had has enchanted us, has fascinated us, and has left us in a state of mesmerism and hypnotism. I therefore convey my heartiest thanks to him. I, I hope her